Hi everyone, my name is Jamie McClellan. I am going to be speaking to you today about my poster on the characterization of immunodeficient mouse models that we have available at Envigo. So the purpose of this study that we did was just to evaluate the immune phenotype in some of our immunodeficient mouse models that we have at Envigo. So when we're talking about SCID mice, we're talking about a spontaneous mutation in the Prkdc gene that leads to highly aberrant genetic rearrangements that impair the function of T and B cells by not allowing them to generate T cell receptors or immunoglobulins. However, some SCID mice, according to the literature, about 2 to 20%, randomly produce functional rearrangements in the DNA, allowing for functional T cell receptors or heavy chains for antibody production. These cells can actually go on to survive and expand, and leading to the production of a measurable serum IgG, and these mice are considered leaky. So the methods that we used for this study were blood spot collections, dried blood spot collections at 12, 16, 20, and 24 weeks of age. Those samples were sent off to IDEX Bioresearch for MSI analysis of IgG. We did necropsies at 6 months of age to look for the incidence of thymic lymphoma, and we started with an N of 23 per strain. So the first strain that we looked at was our CB17 SCID, and we have, in the dark gray bars are the males, and the green bars are the females, and we have IgG measurements, the percentage of animals that were over 5 micrograms per mil of serum IgG from 12, 16, 20, and 24 weeks of age. And then the next strain that we looked at was our CB17 SCID with the list beige mutation, which reduces NK cell activity. And then the next strain was the SCID model on a NOD background. And then we also looked at our SHORN model, which is the NOD SCID with the hairless mutation crossed in as well. And if we look at all of this data together, it just shows us that our animals confirm with the literature saying that 2 to 20% of SCID mice or become leaky over their lifetime. So we also looked at survival in these models and noticed at 24 weeks of age, or about 6 months of age, that over 80% of our animals still survive to 6 months of age, and these are immunodeficient mice. And then with our thymic lymphoma incidence, also confers with what we find in the literature at 6 months of age, with less than 30% developing thymic lymphomas at 6 months of age. So the immune phenotype can vary depending on multiple things, including the background strain, whether it be the CB17 SCIDs or the NOD SCIDs, age as well, that they can increase with age. It can also depend on extra mutations that are on these backgrounds, such as the list beige mutation affecting NK cell activity. A major one is housing environment, which can be considered barrier versus isolator raised animals, which is pretty much just the health standard of the animals that are available. So we still need additional studies to confirm immune cell activity and function, which would be useful to further characterize these models. But these data still provide valuable information to determine the best, to help us help our clients determine the best models for their xenograft research. Thank you.